Hello everyone, welcome to our Ant Biology webinar series. In this series, we generally have fortnightly lectures on weekend. Our Saturday special lectures focus on relationship between biology and other disciplines, and our Sunday lectures focus on uh, various branches of life science. And to our listeners, uh, to our listeners, you can post your questions on the comment section below and continue till the end to know deep about the work that what the speaker is going to say. Hope you will enjoy the talk and get enriched with the talk. Feedback links will be given at the end of the talk. So continue to listen. And now, without further delay, I am requesting Professor Ena Roy Banerjee, our mentor and convener, to introduce today's speaker, Professor Sohan Prabhakar Modak, to our audience. And over to you, ma'am.
Thank you. Thank you, Shahili. To all the viewers today, a very warm welcome. We from the Translational Outcomes Research Group and Immunobiology and Regenerative Medicine Lab of Department of Zoology are very, very happy to be organizing this webinar series. As Shahili said, we do this fortnightly, but this time it's an exception because last Sunday, our very esteemed speaker, Professor Sohan Modak, was supposed to give the talk, but as things would turn out to be, the powers that be, right, Professor Modak probably wanted us to do it today. So since there are certain powers which are beyond our control, and which is something that Professor Modak will be covering during his lecture today, the wonder called life, and the little the cogs in the wheel that make it turn. And so in ant biology, the symbol ampersand is very, very loaded. In biology, we believe that something that celebrates life cannot be understood in its entirety only from the people who practice strictly biology. So, as Shahili said, by way of introduction, Saturdays are dedicated to speakers who speak not strictly on biology, but maybe from another discipline, from another perspective, and deal with certain subjects in biology. But on Sundays, like today, our speakers are biologists. They tend to discuss, reflect upon, and sensitize us about the various developments in the fields of biology. And today it is my extreme honor and pleasure to introduce to you Professor Modak, who is our speaker. So I must apologize that this is something we cannot control. I think you can also probably hear the musical procession going close to my house, and this is something I keep saying that, like the internet, like the digital format, this is something we cannot control. Is the environment in which we sit. Like in a conference theater, we might have other sounds coming in. You will have to excuse me, and somebody is having some revelry and passing by my house, so you will just have to ignore that music. So a very warm welcome to our speaker. Thank you, Professor Modak, for agreeing to speak on this platform, and I'm honored to read from your very illustrious bio sketch, which is going to be very, very brief. So, Professor Sohan Prabhakar Modak, an embryologist and evolutionary bioinformatician, did his PhD from the University of Geneva and his postdoctoral work at the Oak Ridge National Laboratory and the University of Kentucky, Lexington, USA. He served as staff scientist at the Swiss Institute for Experimental Cancer Research from 1970 to 77, as visiting professor at the Ohio State University, and as scientist engineer at the French Center for Nuclear Energy, Grenoble from 1978 to 1979. In 1979, he joined the University of Pune, currently known as the Savitribai Phule University, as professor, and he also served as the head of the Department of Zoology there. Professor Modak was Professor Emeritus at the Karnataka University,
I'm on webinar. I'll
Dharwad,
call you later.
from 2001 to 2003, and the G.N. Ramachandran Senior Research Fellow at the Institute of Genomics and Integrative Biology, that is IGIB, New Delhi, from 2005 to 2008. He founded the Biotechnology Training Program NCCS, Bioinformatics DIS, as well as the Department of Biotechnology and the National Facility for Animal Tissue and Cell Culture, or NCCS, as it is known now. His research interest lies in the field of embryology, classification, and evolutionary 3D tree building. His notable discoveries include the demonstration of the degradation of chick lens cells during normal development apoptosis and molecular phylogeny. And I was just reading your little discourse and that the word apoptosis, and I hope you will be talking about that at the end of your talk. And molecular phylogeny in 3D. Professor Modak's research has been published in numerous journals of international repute. He currently mentors students on molecular evolution. And that by no means does justice to the illustrious personality that we have tonight with us. So, a very warm welcome and over to you, Professor Modak.
Thank you. Thank you. What I'm going to do today is to very briefly explain first what we already know about the living systems, especially eukaryotes or prokaryotes. Basically, are composed of living cells which contain four major biochemical molecules, and the information that how to make them and how to reconstitute or compose them into a living entity. So here you have a picture of, say, showing the cells, and then if you open the cell, there is a nucleus, and the nucleus is full of this noodle, which you pull out the noodle. It's a string, is a chromatin, and the chromatin, when it's further unwound, give rise to the long string of double-stranded DNA. The double-stranded DNA, of course, is, as you all know, is not a naked molecule, but it's complex with nucleoproteins, which protect it from a variety of physical damages, and also protects regions of it from even contact with chemicals per se. And this is the issue that we're going to deal with: genome, and the stability of this DNA molecule is very important for living state. The next slide. Genome integrity and stability are the issues which are fundamental to the living state. Uh, you can see that basically the higher the differentiated state in the system, greater the retention of the stability that is required to maintain its in functional space and time. Stability can be achieved either by redundancy, so that if your part is lost, you replace it by the one that's in the stockpile, as well as from repair by the repair of the damage, which naturally challenges the redundancy. So these are the kinds of issue, because cells are constantly subjected to a variety of damages, physicochemical agents, and DNA is the one which is the major target that matters. Because if the rest of the cell is damaged, then either the damage can be repaired, or of course the cell may die. But if the DNA is damaged, the cell may not die, but may undergo mutation, become dysfunctional, or may even become express disease condition. That's why the restitution of the damage in the DNA to the normal state, without allowing any mutation to occur, becomes very important. And this is something, a process, which is ensured in the living systems by presence of enzymes, catalysts, which can repair, which can detect the damage in DNA and can repair them to restitute the damaged DNA to the original condition. Now, there are different types of physical agents and chemical agents which cause different types of damages. For example, next slide, please. You have um, you have the to understand this, one has to realize that something about the DNA functionality, why this type of nature of the damage and its repair matters. We know that DNA is a polymer of ATGC, forming a linear array of genes that alternate with non-coding regions containing signals to denote type and specificity of each gene. Now, each gene is recognized by a combination of signals located in the regions flanking. The reason talking about flanking region is because in human DNA, only 2% of DNA is genes, and the rest 98% doesn't have genes, but out of that, about 2 to 3% contains regions which are flanking to the upstream and downstream of the genes, which may contain signals that allow recognition, gene specific recognition, uh, so that anytime a particular gene is to be called upon to express itself, then that specificity and that specific signal is required to be activated. So, remaining 95% DNA, people call it junk DNA. Actually, I don't like this term. It's actually stupid because just because we don't know what it does, they call it junk. Because there are questions, is it really junk? Or does it include the regions that can form new genes? In fact, we do know that lot of so-called non-coding region contains segments which are translatable into small, short polypeptides. And who knows, by genetic recombinational processes, one can create larger polypeptide sequences. Anyway, there is another issue about this, and that contains what is the other function of this 95% DNA? Well, remember, if you if you decide to shoot on a target with a gun, then on the target there are many places. So if you want to really reach the center of the target, before you actually do it, you have a lot of hits and misses. And this is exactly the issue, that because the genes are flanked by a lot of non-coding DNA, the damaged DNA damaging agent that damage it may actually be doing hit and miss, so that this allows protection of the gene, statistical protection of the gene, by the fact that there are many lot of non-coding DNA around it. The next slide, please. Okay, so DNA is sensitive to damage by a variety of physical chemical agents. And so what I'm going to do is talk about types of damages that take place in DNA and way those are repaired, and then the biological implication and significance in our living systems and the lifespan of individuals. For example, ultraviolet radiation, which is a part of our sunlight, ultraviolet radiation causes damage to DNA called pyrimidine dimers. So if you have paired pyrimidine next to it, dinucleotides, TT or CT or TC or CC, then UV may cause dimerization within them, and this is a very serious kind of uh, dysfunction that is generated, and it has to be removed. So there exists excision repair enzyme, which actually there are enzymes which are crawling over the DNA. They find the damage, they make an upstream cut, then they make a downstream cut, pluck off the dimer, the pyrimidine dimer, and then comes an enzyme which is the repair enzyme. So it comes in, it starts using the undamaged strand as the template, copies it properly, and rejoins the ends. And this is excision repair system. And excision repair system is very well studied in ultraviolet radiation repair. Then there is also a you can also directly reverse the base damage that is caused because single bases may be damaged. For example, you may just throw off the side chain or you can demethylate the particular base and things like that. So there is a possibility of direct reversal of base damage. You also create apurinic acid or apyrimidic acid sites, and these are removed also by enzymes called glycosylases. Next one. The next slide, please. Yeah. Then you got strand breaks. Ionizing radiation, for example, just break the phosphodiester linkage. It can break phosphodiester linkage in one strand, calling single strand break, or in both strands or double strand break. Single strand break repair is quite easy because it's just all you need is a polynucleotide ligase which comes in and joins the strand break. Double strand break is a more tricky one, and quite often it may lead to the double strand break may lead to two strands, for example, moving away from each other. And if they move away from each other, they don't meet. And if they don't meet, then it's very difficult to actually join them. As long as they're held together by chromatin and positionally, then it's possible to join the double strand breaks, although through a quite complex process. There is also issue of homologous recombination, and for example, there is a disease, Heteroderma pigmentosum, which has a series of problems, especially generated through UV damage primarily. And there is a multi-component system that allows the repair. Next slide. So what we did over years, we developed three different techniques. Uh, Two techniques are at the cellular level, one technique is at the biochemical level. The first technique, I was working in Oak Ridge National Laboratory, and there I met Fred Bollum, who actually invented the calf thymus DNA polymerase, eukaryotic DNA polymerase, which is actually a replicative enzyme. Well, Art Kornberg didn't like him because Kornberg was working on bacterial DNA polymerase and claimed that to be replicative. Turns out Kornberg enzyme is a repair enzyme and Barlam enzyme is actually replicated. Whatever. I had an opportunity to work with Fred, and we developed a technique because I was working on lens of the eye, and in the lens of the eye, I discovered that as the lens fiber cells differentiate, they finally exhibit a phenomenon of programmed nuclear death. That is, fiber cells are formed elongated due to synthesis of lens proteins, and once enough lens protein is made, the DNA is no more required. So the just stacks bodies, dead bodies of fiber cells remain, and the nuclei die in situ. And this process actually is the real program cell death. You will see in the picture next that you will have cells actually disappearing in a very positional manner, and the wave of cell death disappears. So I was interested in this phenomenon. I did a lot of studies on it and need not worry about it right now. But more important, I first did UV ultramicroscope spectrophotometric analysis to demonstrate that as the cells undergo pyknosis, they progressively lose DNA. Okay, this is reasonably easy. Nowadays, it was quite a difficult proposition then. But having showed that, I was interested in finding out why and how does this process take place. So Fred Bollum had the DNA polymerase. So I took lenses, I took fixed the lenses in ethanol because ethanol does not denature DNA, and then embedded them paraffin in low temperature paraffin, 50 degrees. That does not denature DNA. Then made paraffin sections, then deparaffinized such sections on the glass slides, and build the small glass wells around this, just around this section. Fill the well with calf thymus DNA polymerase and appreciated radioactive, that is, deoxyribonucleotide triphosphates, and other things like magnesium, manganese, whatever the divalent cations are required. Close the slide with a cover slip, this well with a cover slip, incubated it for at 38 degrees centigrade for whatever period of time I required. Then I opened the slide, washed off the medium, then treated the slides with trichloroacetic acid in order to remove all the unincorporated uh, nucleotides, and then did radiotography. And when I did the radiotography, I suddenly found that something very interesting was happening, that these nuclei were incorporating DNA radioactivity. The next slide, please. Okay. We can move that next slide, please. Okay, here is a picture of the lens development. Is the top is the lens vesicle is just formed very early embryogenesis, chick embryo, about 3 days of 3.5 days of development, and by the time then it becomes 8 days, 13 days, and what you see, the little blackish shadowy thing in the middle of this white ball are the nuclei, and the nuclei have disappeared. It's unfortunate that this picture is kind of not really high resolution picture. Nonetheless, in the middle, the nuclei have disappeared, and you will see that the nuclei actually have undergone pycnosis or degeneration. Next slide, please. Okay, here is the picture now. Is the lens epithelial cell nuclei, radioautograph, and then they incorporate a little bit of radioactivity because they contain template DNA. So anywhere there is template DNA, if it's denatured, then it will become a template, and calf thymus DNA polymerase can recognize it and carry out in situ incorporation in chilled fixed cell nuclei. Here are on lower half of the lens fiber cells. These are elongated fiber cells. They're still normal ones, producing synthesizing lens crystalline messenger RNAs, and of course, which will be converted to lens crystalline. And then towards the middle, the nuclei become vacuolated and they become fragmented, and they've become very small. And now you can see, as they're becoming smaller, they incorporate far more radioactivity than the ones which are on the epithelium or the normal fiber cells. So this is despite the fact, therefore, the nuclei which have the smaller amount of DNA increase show increased incorporation with DNA polymerase. That is because DNA polymerase finds many more broken templates in the DNA of the degenerating nuclei. So what we are really looking at is increased strand breaks in DNA occurring. So we postulated, therefore, for the first time, that this is not only programmed cell death, but this programmed cell death involves actually progressive accumulation of single and double strand breaks. Until then, the whole fragmentation process leads to loss of nucleus, which was, of course, a confirmation of our ultraspectrophotometric analysis. The next slide, please. Then we use another enzyme, which maybe you're familiar with this, which is commonly used in an assay. It's called, also invented by Fred Bollum, and that is a calf thymus terminal deoxynucleotide transferase. This is an enzyme which actually uses only one nucleotide at a time as a substrate. It doesn't like four, it takes only one, any one of the four. And what it requires is a free 3' hydroxyl end. So if you have free 3' hydroxyl end, like in what I'm showing here in my finger, okay, maybe this way. Okay. Then it will attach single nucleotides by end addition reaction. And this is a very rapid synthesis, so that if you have single strand and free single strand ends, it actually can massive DNA synthesis, end addition synthesis occurs. And when that was done, then we could show that nuclei undergoing pycnosis are very heavily labeled, while the corresponding amount of label is substantially less in normal lens epithelial or fiber cells. And we did kinetic analysis, showed what you do in this experiment is that you take the same sections, but depending on some sections you incubate for 1 minute, some for 5, 10. So you can do a time course of incorporation using terminal transferase. And then you can plot by counting the silver grains, you can plot number of silver grains as a function of time. And we could show that actually from the slope of the curve, many more single strand breaks were generated during the nuclear degeneration. I am using this term programmed cell death because when I actually did this first work, there was only one other example of such localized programmed cell death, and that was during the digit formation in the embryogenesis. When the digit embryogenesis limb is formed at the tip of the limb, it's a mass of cells. Then progressively, at the position of the digit, cells undergo death, programmed death, so that that region separates out into the digits. And this was done by Saunders. And just 3 years later, I did the work on the lens, but I showed this. And it's only much later that people found similar phenomenon elsewhere, and they said, well, they want to become hotshots, so they call it apoptosis. Now, it's a very bad term because apoptosis is a Greek term. In Greek language, it means falling off the cliff. Well, falling off the cliff is a suicide phenomenon. You jump off the cliff or you're thrown off the cliff. Actually, that happens with necrosis. When the cells are extreme conditions, then the cells explode or die in situ. They become necrotic. And necrosis is not the same as apoptosis because necrosis can happen anywhere, the first position of contact with the deleterious agent. Anyway, we'll leave it at that. The next slide, please. Okay, then I developed another technique now. Of course, this is a classical technique of agar gel electrophoresis. You can see on the left-hand side panel, I took the lenses, then dissected different parts of the lens. So fibers, which are young fibers, I extracted DNA from young fibers at different ages, just 5 days, 8 days, all the way up to 19 days embryogenesis. And I loaded them in agar gels, purified DNA, and electrophoresis. And lo and behold, all the DNA gets stuck in the well because it's all high molecular weight DNA, because most of these nuclei are very active. Well, when I did that to the central region of the fiber where the programmed cell death was occurring, you can see that there is a 8 day. There is most of the DNA is on the top. You can't really see much before. Then there comes the track 13-day lens. There is some DNA in the top, high molecular weight. And now you see fragments of discrete size. 1, 2, 3, 4. You look at the 15-day, even more discrete size fragment, 1, 2, 3, 4, 5. And same thing as 19 days. I must tell you that these gel strips done for 8 days, then 13 days, and 15 and 19 days were different experiments. They were collated together. That's why the distribution of molecular weights is different. But nonetheless, the objective was to demonstrate fragments. Now, this is very, very interesting because this is exactly the time when the chromatin was demonstrated to contain a nucleosomal structure. And every nucleosome contains DNA molecule, which is roughly 200 base pairs, varying among species. But and that is called as a monomer, dimer, trimer, tetramer. So actually what we're seeing, mono, di, tri, tetra, pentameric fragments of DNA. So the objects, what this says is that during programmed cell death, DNA is broken down in such a way that you have fragments which are which are fragmentation of the chromatin. And next slide, I developed the technique for two-dimensional electrophoresis. The next slide, please. Okay, here what I did was that I first take the slab gel. I did only one track at the side, which I uh, put the DNA, which was purified double-stranded DNA. I electrophoresed it, and don't look at the top, the bottom slide. And when you do electrophoresis, the bottom left side, you can see the horizontal track up you can see mono, di, tri, tetramer, and some material in the high molecular weight region. And then I took that particular gel and soaked it in alkali and turn it at 90 degrees and then electrophorese it at 90 degrees. And when I did that, then those fragments come down and along the diagonal. So you can now see single stranded fragments equivalent to mono, di, tri, tetra. And the high molecular weight DNA stays. This is what happens. So that most of the strand breaks are actually double strand breaks, are very close to single strand breaks in the internucleosomal region or linker regions. Okay, so this technology is very useful because that was used subsequently to study. One of the issues is that what happens during the process of aging, because after this work was done, uh, I worked in Swiss Cancer Institute, and then I joined Ron Hart's lab in Ohio State for a short period of time. And Ron Hart had done very interesting work on the aging of the mice, and especially the issue of caloric restriction. The point is the following: if normal animals in the wild, uh, their lifestyle is accustomed or adjusted to their ability to find the food, and depending on the food they find, sometimes find a lot, sometimes find a little, and so there's no regularity between their food finding and food intake. When they find a lot, they gulp it all a lot. When they find a little, they starve a bit. And so there is no regularity. And the question was, is this one of the parameters important in lifespan? Now, there is a place in the United States, San Diego. When San Diego is one of the most beautiful zoological gardens, and this zoological garden is also very famous. I've been there, by the way, because it's a 125-year-old garden, and very nice, clean records have been kept of all the animals. Each animal, records were kept on its daily behavior, how long, at what age it was procured, was it born there, was it taken from outside, and when did it die. So it has lifespan information on a variety of animals. And this is the best data available in the world as far as the lifespan of mammals is concerned. And so what he did was, having done and looked at, he established a laboratory for mice, and then it was C57 black and A crosses, and he put them in a laboratory conditions and did decide to do caloric restriction experiment. A caloric restriction experiment is such is that if you leave the mice in the laboratory and give them food, they eat all the time. And whenever food is available, they eat like the wild animals do. And so they actually are overloaded with food. So normally caloric restriction experiment involves the following. What you do is that during the day you turn the lights off. During the night you turn the lights on. And only during the night, at two particular specific times, you supply food. So the slowly mice get accustomed to this, and they eat food only in a time dependent and position dependent manner and availability dependent manner. This is a dramatic effect, and it was noticed that this phenomenon leads to 30% increase in lifespan of mice. And this was the model I wanted to use. Now, Ron Hart had done this experiment, but he has done another experiment preceding this before he was also in Oak Ridge National Laboratory. He cultured human cells like Hayflick has done. Hayflick was the first one who cultured human embryonic fibroblast cells, cultured them in vitro, and when he passaged them, he found that after 65 passages, the culture died. And he invoked the principle of human cells in culture exhibit finite lifespan or finite number of cell division cycles. So Ron actually took such cells, grew them in vitro in conditions of finite lifespan conditions, and at different times during the lifespan, he irradiated them with ultraviolet radiation and then gave them a pulse of tritiated thymidine in the absence of UV, and then found the incorporation, which is equivalent to excision repair. And he could demonstrate that the cells immediately underwent excision repair. However, the amount of excision repair varied between the time of lifespan. This is something I'll show you later. It is relevant to what I said just now, but it'll come a little bit later. Next slide, please. I wanted to look at another system to apply this problem of localized incorporation in situ labeling method of determinant transferase. And during uh, the reproductive cycle of the mice, they undergo, if the female mice undergo the standard every month, they undergo the problem in response to the hormones, and you get in a pseudo pregnant type of scenario, the vaginal epithelial cells begin to exhibit programmed cell death, and keratocytes die. So we took the keratocytes and sections of the vaginal epithelial cells, vagina basically, and incubate them with terminal transferase. Now, there are two ways you do the experiment. You take the ethanol fixed slides and do the treatment, and then another case as a control, you treat them first with denaturing agent and then give the enzyme. So when you denature them, more strand breaks become exposed. You can see the top one is vaginal section, the epithelial cells on the right side, and they're undergoing pycnosis in pseudo-pregnant mice. They're labeled. But when you took the acid denatured ones, they label even more, much greater amount of labeling occurs. This simply is a control for the experiment. The point of the exercise is that even during the programmed cell death in vaginal epithelial cells, you get accumulation of strand breaks, which are appearing prior to the loss of the cells. The next slide, please. So, terminally differentiated and aging postmitotic cells accumulate lesions in DNA due to defective repair system. That was what I postulated in 1972. And now there's enough evidence has gathered over, I said 30, but actually 50, 60 years now, 50 years now that this is true. So nuclear degeneration during programmed cell death and appearance of repeating class of low molecular weight fragments that are related produce differential digestion in vivo of the lens chromatin. Now, it is suggested that the chromatin structure controls the accessibility of damaged DNA sites to repair enzyme. Thus, chromatin structure as well as DNA repair enzyme together control the maintenance of genome integrity. And also significance of repair potential and cell proliferation potential, the latter delimited by Hayflick's experiment, determine the stability of differentiated state. So I've been interested in the process of what is the stability reason for a stable differentiated state. For example, a fibroblast cycle. But in the lens of the eye, for example, or muscles where myoblasts are converted to myocytes, or keratocytes, etc., cells undergo all kinds of irreversible changes, and they become terminally differentiated, that they reach a point of no return with reference to their ability to divide. And in that position, the question is that whether this is true also during the aging, because in human tissue or mammals, for example, 99% of the cells, 99.9% of the cells in adult do not divide. Only 0.1% of the cells remain, can maintain the division potential, or as a stem or founder cells, which can multiply and do the function of repair, replacement of the damaged cell. So most of majority of cells do not divide. They do not because they have reached a point of no return. Next slide, please. So we went back to this uh, experiment where I established a colony, very beautiful colony, um, mouse colony, and then we took 3-month-old mice. We isolated cerebral cortex, embedded them in paraffin, and did tissue section as I described before. And then also we took 30-month-old mice and also did the same tissue, uh, fixed ethanol fixed section. And then we also took 3-month-old mice. We first irradiated them with 20 grays of X-rays, and then fixed them and took the cerebral cortex. So you have three samples, two samples, 3-month-old mice, 28th, not 30, 28-month-old mice. You have sections which are ethanol fixed and of course unirradiated. Then you are X-ray irradiated, ethanol fixed and X-ray irradiated. And then you have sections which are uh, ethanol fixed and then acid treated to denature DNA in situ. And now these sections were incubated with cap-thymus DNA polymerase. You can see on the left panel the 3-month-old mice incorporate a little bit of radioactivity, X-ray irradiated, many more silver grains, and the acid denatured ones in the 3-month, many more silver grains, because the single-stranded DNA breaks, which are now revealed by denaturation of DNA. That happens comparing the top 3-month-old undenatured to the bottom 3-month-old denatured slide. On the 28-month-old, you see many more strand breaks, much more grain count, even in undenatured condition, and much more after X-ray radiation, and even more after in unirradiated 28-old ones after acid denaturation. This is shown in next slide quantitatively. Next slide, please. Okay, here's, because I think the time is going to be limiting, so I will quickly go through the slide. You can see this is what we do, then the grain counts per nucleus in the unit nuclear area. And you can show that unirradiated cells, which are undenatured, mean grain count is about 8.9, let's say 9 grains per nucleus. Undenatured and unirradiated brain section, then you get 22.3, and you get X-irradiated ones, that gives 24.6, and you said acid denatured, and you see much more. So you get dramatic increase in the strand breaks in during the process of aging in the cerebral cortex. Actually, I should mention the cerebral cortex contains 6 cell layers. The first cell layer that is formed during embryogenesis is called number 6. And then comes number 5 and number 5, because the number 1 is the most exterior one, and number 6 is retained towards inside. So you also see the cell layer dependent changes in the aging process. Basically, the next slide, please. What this is showing is that, next slide, we also measured the methyl residues, 5-MC residues, by an immunoprecipitation reaction. and you can show that as a function of age in the liver cell nuclei, the amount of 5-MC levels decrease. This is 5-MC level from DNA and not chromatin. Remember that. Purified DNA and you isolated the 5-MC. The next slide, please. Okay, then we look at the chromatin itself. Here is the total lifespan of mice, starting from newborn mice. This is actually 2 weeks postnatal liver, then going to 1 month, and from 3 months to 26 months is a flat curve, and then it goes up. What we are actually looking at, we're isolating nuclei, digesting them in with micrococcal or staphylococcal nuclease, and then looking for acid perceptible DNA in a limit digest. So what happens is that whatever the DNA is accessible to this enzyme is digested off, and the rest remains undigested. And what you're really looking at is stable chromatin regions which are not accessible to micrococcal nucleus. Lot of this DNA, about 70% of the region is accessible in a very early embryonic, postnatal stage. And by from about 3 months to 26, 25, 26 months, 50% of the chromatin remains uh, um, resistant to micrococcal nuclease, and then this resistance increased up to 63% during the last 20% of the lifespan, or 15% of the lifespan, to reach 63%. Remember, if you have nucleosomes of DNA with 200 base, if the entire DNA is covered with nucleosomes, then roughly about 70% of the DNA is covered by actually histone particles, and the 30% is in the linker DNA. So maximum amount of DNA that you can digest when entire DNA is covered with chromatin is 30%. So 75% is maximum that will reach, and actually we're reaching that level almost by the time end of lease. So what we conclude, therefore, that aging process involves not only accumulation of DNA strand breaks, but also uh, uh, inactivation of chromatin, its condensation and accumulation of some. If we measure the amount of histone, the actual amount of histone does not increase. This is surprising, but that means that there must be free molecules of histones in the cytoplasm which has come off. There is an alternate explanation, and that was by Weintraub. Harold Weintraub suggested that some nucleosomes exist as half nucleosomes, and they remain attached to DNA. Half nucleosome, that means H2A, H2B, H3, H4 histone only at a time. In a nucleosome, there are dimers of these. So in a half nucleosomal DNA is completely sensitive to micrococcal nuclease, while in a full nucleosome it is resistant. And maybe that's what's happening. The next slide, please. The next slide. So I think we will just, this is what I have said, so you can go to the next slide, please. Okay, here's the excision repair phenomenon. This is about the last two slides now. What Ron Hart did actually was measured the amount of excision repair in primary cell cultures at different ages. So what he did, he took, for example, cultures from shrew, going to the mouse, then the rat, hamster, uh, cow, elephant, man. Actually has done 20 more other species, but I'll show this as an example. You can see that as a function of time, what did I do? Take the cultures at the same age after primary culture, then hit them with the same dose of ultraviolet radiation, and there, by the way, the cells are in the same concentration on the plate, same amount of UV is given, and the same amount of radioactive isotope is provided in the culture conditions. The culture medium is the same, and then you allow at different times, you take samples to find the amount and fix the nuclei and find the amount of silver grains accumulated. So what this plotting is, the average number of grains per nucleus is a function of time. And oh, lo and behold, in the mouse shrew, for example, the plateau reaches much faster. Then comes the mouse rat, and it's much higher incorporation in man. And what this was said, then when you take this kind of data, plateau positions, that is maximum incorporation in man, elephant, cow, and also chimpanzee and gorilla, and all these animals were done, and plot such a silver grain data, plateau silver grain data, against the log of the known lifespan as was recorded in San Diego Zoo, you find a straight line, semi-log plot. What this directly means is the repair potential, at least UV repair potential, is directly proportional to the log of the lifespan. So if you have more better repair, you will live longer. And this is probably what caloric restriction does, is that it ensures the improvement in your repair potential. The last slide, next slide, please. Okay, we'll last point is that Hayflick's story was further recorded, looked at, uh, because we said people said, well, the cells are dying after 65 passages. In mouse, they die after 55, and so on. Why? Why do they die? And there might be some problem with DNA. And turns out, at the end of each chromosome, there are small fragments, small regions, which are called telomeres. Telomeres because at the end of this. And there are multiple copies of telomeres attached one after another to the end of the chromosome. And such telomeres are very interesting because if you remember, if you want to replicate DNA, then you must have a starting point. So what happens is that when the two strands separate, what happens in the middle, we already know there is a different type of scenario. But at the ends is a very serious problem. So what happens is that the 3' hydroxyl end of the chromosome of the DNA turns around, forms a hairpin, and at the hairpin you got 3' hydroxyl end, and then the DNA polymerase can move up and start replicating in the 3', 5', 3' direction. And therefore, these telomeric fragments are very interesting because they turn around, form the hairpin, and then they allow DNA synthesis to take place. And after the DNA synthesis, apparently, they're cleaved off. So a telomere is cleaved off. Every cell division, one telomere is cleaved off. So if depending on number of telomeres you have, number of cell divisions you have, unless you have a possibility of an enzyme called telomerase, which can restitute them. And if there is any defect in that, then you can generate a problem. If you have too much telomerase or mutated telomerase, the cell can just keep on growing like crazy and multiplying, like happens in cancer cells or in aneuploid cells. I think I'll just conclude saying that, therefore, these telomeres represent a clock, telomeric clock, and this telomeric clock decides the Hayflick limit, that total number of finite number of cell division cycles, and that these two together are dependent on the ability of the DNA repair mechanism, and the total lifespan dependence is on these three factors: the repair system, the telomeres, and number of cell cycles that are possible. Now, there are many other implications of this which I will not talk about unless there are any questions. The simply conclusion, the genomic stability is actually maintained by presence of repair enzymes, and actually it is relevant to the telomeres in terms of the genomic stability for the lifespan determinant. Thank you. If you have any questions. Okay, that was the
Thank
last
you,
slide.
sir.
Right.
Thank you, sir, for this wonderful lecture. And
Thank
we
you.
have some questions.
Yes.
We have some questions. Can we go through them?
Yes, yes. Please do. Oh,
And
the DNA strand. Yeah, the DNA
our
strands
first,
break. The first question is, why do
yes,
DNA strands
why
break?
do DNA strands break?
Right. The DNA strand breaks because the 3 prime 5 prime bond between the adjoining nucleotides is very sensitive, and it's the one which is affected more frequently. Of course, if you have X-rays or ionizing radiation, they find this place is the most sensitive. Naturally, the ionizing radiation can also affect the purine ring or pyrimidine ring, but that will cause a purinic acid flip of the purines or pyrimidines. But not most obviously, even in that case, this will lead to a strand break. So DNA strands break because the physicochemical, the say, covalent linkage, that linkage is broken rather readily. Next.
Now the second question comes, for
Yeah.
mutation of P53, is there any chance of DNA repair?
Yeah, there is a chance of DNA repair, but it's much more difficult proposition. Because you depends on the listen, I told you there are a number of types of DNA damages, and there are different types of repair systems. So depending on the type of the mutation, I mean type of the DNA damage that is affected, the P32 mutation will come in, because P32 mutation does not really is not really effective in all necessarily all kinds of repairs, but some kinds of repairs more than the others. So it depends the type of the damage accompanying mutation of the P53. Whenever the kind of damage has no relationship with role of P53, then of course it will not be affected.
Okay, thank you, sir. Our next question from Nobbendu Mukherjee. Can we destroy cancer cells by attacking it with zero-dimensional molecules or by tagging it or coating it with bio-nanofilms by preventing it to proliferate further?
Yeah, actually, this is what people are trying to do that right now. The problem is that you need a targeting system. For example, people try to get nanoparticles which are coated with an agent which specifically bind to DNA or bind to the cell surface, which has a specific antigen, like a cancer antigen on it. And then the particular nanoparticle with the antibody will attach to it and can target such a cell specifically. Because in vivo there is the issue of coating it does not exist, because there's a random distribution of cancer cells. When it's in a tumor form, then it's a localized scenario. But when you got metastasis, the cancer is spread in large number of spaces. So this technology of nanoparticle coating with an antibody in order to target it towards a cancer cell carrying the corresponding antigen is now being looked at very seriously by different laboratories in different parts of the world.
Okay. Our next question from Akashlina Marik. Does genomic stability has a role in disorders related to metabolism?
Yes, it is, of course. That's what the one of the example was, of course, the xeroderma pigmentosum. Uh, Directly, the disorder is its inability of excision repair, and therefore you accumulate strand breaks, and the person dies very early. The children died between any time between age of 8 to 15 because of xeroderma pigmentosum, a disease very prevalent in large scale in South Africa, especially with the regions which have a huge amount of and actually the genomes of Scottish people, for example, at least that's what was shown the other day, many years ago, was more sensitive because they have much reduced excision repair system. So they cannot readily. I'm not talking about all of the Scottish people, but the mutation is prevalent among the Scottish people. And this mutation leads to uh, disruption, this, this, this disability to repair damage. Yes, so the has a role in disorders.
Okay, sir.
Actually, it's the other way around. The disorders are due to the stability. Yeah.
Okay. Thank you, sir. And next question is from Mandhotra. Sir, we are studying these experiments in vitro, but what about system biology?
I don't understand the question. I understand that you are studying experiments in vitro, but what do you mean by system biology? Because if you mean by in vivo scenario, but then I described the lifespan potential, that the lifespan of mice, for example, total in mouse colonies, is that what you will call it system biology, or what do you mean by system biology? I need to understand the question because that is a system. Unless the person needs to, Manish, you want to ask, you want to clarify the question. Meanwhile, you have another question.
Another, yes, sir. So next question from Bijay Biswas. Is there any significant effect of anti-aging cosmetics on genomic stability or on aging at all? If yes, then how?
Okay. So there are cosmetics, for example, which are used. Women use that quite often, going to the when they go to the beach to undergo browning. Okay, they're affected with ultraviolet radiation, and therefore these cosmetics, anti-aging cosmetics, lotions, for example, they contain UV repulsive agents, and consequently they do not affect the skin, do not create skin or protect the skin against any kinds of disorders created by excessive ultraviolet radiation. Okay, and naturally that will affect the genomic stability. It will help retain or maintain the genomic stability. That is a common anti effect of anti-aging cosmetics. These are not really anti-aging per se, but they are anti-UV cosmetics. Uh, I don't know about any anti-aging cosmetics which have really been proven to be anti-aging, because looking good is one thing, but calling it anti-aging is different thing. Anti-aging means it should affect the lifespan. It should increase the lifespan. Do these compounds increase the lifespan? The answer is no, so far as we know. But then maybe we have to wait 100 years to actually get enough data to make that statement.
Okay, sir. Our next question is any comment on relation between our diet and aging?
Yeah, actually, yes, that's what I was. The caloric restriction actually tells that yes, there is a relationship between diet and aging, and it doesn't say whether you are non-veg or vegetarian. What it says is that the how much you eat, and when do you eat, and with what regularity do you eat, or do you simply is hogging all the time? And do you eat excessive amount of time, or eat in smaller portions at a different specific period of time? For example, some people claim that you eat 8 times a day. That itself is not a solution. You eat a lot of eat twice a day. It's not a solution. Actually, the caloric restriction experiment shows that actually one has to eat just enough, maybe 1 or 2 times a day. This is for mice. I don't think this particular scheme has been well established for humans as yet. But answer is yes. Diet has the amount and the timing of diet intake does have something to do with lifestyle.
Okay, sir. These are all the questions from the comment section, and
Okay.
thank you for your all explanations. And
Well,
now I will. Anything
yeah,
you want to say?
yeah, I just wanted to thank you, Shaili and Vijay, also for helping set this up. Thank you. Yeah,
And I will now request Professor Enaroy Banerjee to give the ending note.
right. Thank you.
It was such an enjoyable lecture. Thank you so much, Professor Modak.
Thank you.
Thank you, Shoheli, also because I think Professor Modak and our Shoheli has been communicating over the last 2 weeks on and off. So
Thank you, ma'am.
this is a rewarding culmination because it was such a wonderful lecture. Professor Modak, aside from the scientific discussion that we were just privileged to hear from you, um, what would be your I was again revisiting your teaching is reaching, and I was hoping that you would say in terms of comments, suggestion, observation to the teachers, because right now with the lockdown and the new normal and various pros and cons in the new way of teaching, teachers would like to hear from you what your take on it would be.
Yeah, well, there is the one issue is the following, and I'll tell you one of the merit of offline teaching. What, the most important merit of the offline teaching is that the teacher and the students, collection of students, have a eye-to-eye contact directly all the time. And for example, when I was teaching, I will look at every student, not necessarily in the same order, maybe a random order, but I will. And when I look at the student is playing games with me. Either the student doesn't want me to look at him or her, so he'll look down. We'll play games as if show as if he's writing something, or talk to someone next door to on the next bench, or actually look at me and establish the contact. So this is one of the games that the students play. Okay, and this this is one issue. Second issue is that when I look at the student who is looking at me, I see if the student has a question to ask. So is there a question mark on the student's face? Or is the student happy with what I'm saying, or has seems to have understood what I'm saying, or really indifferent to what I'm saying? So basically, that one-to-one contact on an offline lecture is very important, and that is something is happening. Now, I've been doing online teaching for last year and a half now, and I miss this completely in this particular contact. And somehow we need to use softwares which are where you can see a string of student faces on the screen. Normally there is a limitation, maybe 5, 6, sometimes 10, but you can't really see 30 students. And some classes have 50 or 100. So I mean that is out of question. So there is this difficulty in the eye to eye, because student to teacher contact is affected in online teaching. There's nothing you can do about it unless there is a better technology available. Now that's one issue. Second issue is the problem of the teacher and the student. Uh, there are no stupid students, by the way. That's the first assumption you must make. Okay, and there are students who are scared of asking questions because they don't come from English medium schools, or they come from a part of the society where they have been repressed, or they're afraid, so they don't dare ask questions, because if I ask a question, what will the teacher think of me? Maybe it's a stupid question, because maybe they don't know how to articulate the question. So there are these issues that the student face with respect to this teacher, and how to handle that. And I tried to manage that, which you have read my article where I actually did that experiment and I repeat that experiment every time it works out. What I do is that after 5 to 6 minutes or a short part of the topic that I talk in the lecture, whether it's offline or this only offline, but I have not done it on online as yet. And offline is that I stop the lecture about 5 10 minutes of section, and then I said, "Do you have any questions?" And the students, if they have questions, they ask. Most of the time, they don't. Maybe because they understood everything I said, or maybe because they understood nothing of what I said. Uh, so then I'm scared that if they don't have questions, then there's some problem here. How come they don't have any questions? Because while I'm talking, I have questions myself. So then I say, well, I will ask you the question. So if there are, let's say, 20 students or 30 in the class, I start with number one. I ask a question. What do you think of this? The student no, then go to same question is transmitted for 1, 2, 3, 4 to 30 students. Same question, and progressively bits and pieces are added to get an answer. Often at the end of the first round, there is no answer, full answer. Then I give the second round. And if there's no answer, then I said, okay, now you're 30, so we'll make random groups of 5 students each, make 6 groups. Just pick them one randomly and make 6 groups, and each group of 5 students sit together. And I said, I'm going to quit the classroom. I leave you 5 minutes, see if you find an answer. And you know what? They do. In 95% of cases, they talk to each other. They are not from the same social strata. They are not from the same cultural disposition or religious background or for educational background. But now they peer to peer discussion. They talk to each other and they tell each other what it is. And they tell each other much better than the teacher tells them. Okay. And then 10 minutes later, they solved the problem. They got it. 95% of
Absolutely.
cases. That's pretty good. So this
Even
experiment,
in
yeah, go ahead.
even in online classes, there is something called, especially in Zoom and such softwares, they allow something called the breakout room. So
Yeah,
what you're saying is absolutely true. So there are 100 people, say, in a session, and then when in the breakout rooms there are 7 or 8 people, and then there's automatically breaking of ice, and people start talking. But before
yeah,
that, everybody will keep quiet in a large crowd. I think that's a common psychology
yeah, yeah, absolutely.
amongst the students.
I started this practice first in U.S. when I was teaching an undergraduate students, simultaneous to my research condition laboratory, and then I tried it again in Switzerland. Of course, the language was different. And then when I came to India, I said, this is it, because here the students should have no difficulty in English language. US, the English is different. It's a slang. In Tennessee, it was a college of black students, and they're completely different language, or slang is quite different. And there, of course, interactivity with the teacher is totally different than the else places. So I was experiencing, and the experiment worked. Finally, I got a chance to work it in India. Then subsequently, I've been teaching at different parts of the country or elsewhere in outside India, and it works. It works beautifully. Peer-to-peer interactions is very important, and that's what we teachers should be doing: inciting, stimulating peer-to-peer interaction instead of allowing them to form them rival groups of comparators.
Especially in science innovation, something that probably we all have been discussing is that school students, especially in our system of learning, are made to rote learn a lot of the things, and
Yeah,
write whatever is in the textbooks, and that goes from the undergraduate to the postgraduate level. Especially in the new normal, we were all,
that's right.
since it's an open book system, and everybody is copying from the textbooks or whatever, and then
Yeah,
we were all thinking that we should give critical questions, but this
yeah.
criticality has been so missing. So why don't you say, um, what your thoughts are on that. Please
No,
share
I
your
think I
thoughts with
agree.
us.
I agree. This, for example, I'll give you a sidekick example of examinations, online examination. I've done online examination. And I know when I'm doing online examination, the students have their books around and each one copies lines directly because with computers you can do copy paste and they produce a printed answer, copy paste answer, and you can even see that the first two lines were in different font, and then the next two lines and the next third line was not only different font but different size. So they are simply copy pasting, and this does not tell whether they are understood. So generally, the principle of an examination is to test comprehension. And the exam system today is not really, has nothing to do with the comprehension, has to do with memory base. I'm not against the memory, but memory and comprehension are equal, should be equal partners, if not comprehension being much more higher percent proportion than memory itself. Because sometimes you may not remember yet understand what's going on. And so this is a problem in online offline scenarios, but comprehension is an issue that requires really a contact differently. Right now, for example, I have a bunch of students that I connect to routinely. This is not lecturing now. Students are doing bioinformatics projects with me. There are about 10 students doing bioinformatics projects, different types of issues we are discussing. And there, most of the time, it's on Skype, for example, or once in a while, once in 5 sessions, we meet actually at my place with masks on and distancing and all that. But nonetheless, comprehension comes a very important issue that does not take place on Skype, but suddenly some of the problems get resolved when we meet once in 5 times. So that is a kind of, I
The
don't know if I've answered your question rightly. But no.
oh
See,
yeah, yeah, yes,
every student, see, there is a problem also. Uh, are teachers really selected because they know how to teach? No. Are teachers really selected because they know the subject? No, not necessarily. Some do, but not always. So, I mean, there is a teacher standing in front of the students, thinks that she or he is God or goddess, right? And knows in know it all kind of thing. It's absolutely untrue. And so you have a problem with teacher selection process also right now. I don't want to discuss it here because it becomes a socio-political issue. Nonetheless, the point is very clear that every teacher should be, you can select a teacher, person to become a teacher. You can select a person to become a teacher and then teach the teacher how to teach. But that's not the process that we have right now. You select the teacher assuming that such a person, because the selection committee selected him or her, or whatever the rules under which the particular person was selected, it was obvious that he or she knew exactly what he's teaching. It's not true. So these are the structural issues in the system,
yeah,
systems issues. I don't know how to
yeah.
handle this
And
because it's
since
a serious problem.
everything is really a continuum, one thing leads to another. So since you
Yeah,
mentioned this is, socio-political issue, or the way we think, the way we perceive things, down to the science that we do, which
yeah,
brings us to another issue of malpractices in science. So
yeah.
the questions that I have been putting across to you, the discussions that we are having, and I think people who are watching now and people who will be watching later, because your talk will be archived, we are all, probably the older of us, are discussing how we can make things better, and the younger people are very baffled, especially because of the new challenges. So I
Yeah.
think one thing is really very closely related to the other. science perception, scientific temperament, the way we teach science, the way teachers are selected, the way examinations are conducted, all lead to that scientific innovation, which is ultimately what it is all about.
Yeah.
So, what would you like to say for that? That
Well,
failure
listen,
is not encouraged.
I have, for example, UGC started this teacher training workshops. I conducted maybe of 20, 25 teacher training workshops, college teacher training workshop. And you know something, I conducted them in different subjects in genetics, molecular biology, development biology, phylogeny, God knows what. And every time only 10% of the teachers actually uh, were actually convinced of what went on. But then I would say that those are the same 10% good teachers to begin with. The other 90%, they took a vacation for. Uh, they got a 5, 6, or 10-day vacation depending on the length of the training workshop, and they were enjoying having fun. Right now, I'm not complaining against that, but I'm just pointing out what happens. So we need a different kind of methodology to actually approach this problem. And it's not just the simple teacher training workshops where one teacher is acting as a teacher to the teachers kind of thing. Because the same problem comes in. The teacher is afraid of asking questions. Does he think or she thinks that maybe my question is stupid? Or the teacher thinks that his question is stupid. And there are no stupid questions and there are no stupid students. That's the first conviction the teacher must have. Only stupidity comes from the teacher, the way he or she actually expresses herself, and his or her disability in communication skills. So, communication skills are the issues fundamental to any interaction, vocal interaction between the students and the teacher.
Indeed. I was again going through your teaching is reaching, and then when you went to the French Institute and you had to learn French,
Yeah.
and then
Yes.
drawings had to be made, and then
Yes.
you come back to where was it, Louisiana, where the language and the accent is different. Then you come back to Pune, and people are not comfortable with the
Oh
accent.
yeah, I had an American accent, and because I had lived in Kentucky and Tennessee, I used to speak very drawn English sentences, not like the British who speak very fast. So Indians, we think that speaking fast is smart. It is not. Speaking fast is not smart at all, because speaking fast, if you speak fast or slow, as long as you articulate, it's okay. But most of the people in our country, because our language differences, our every language has a particular effect, a long-term effect on one's ability to articulate in different language. Consequently, uh, there is a deformation that takes place of the English language when we all of a learn. The way South Indian will speak the English language, or the Bengali or the Marathi or Punjabi will speak, and the big differences. In fact, within our country, we have difficulties sometimes in comprehending. So this issue has to be straightened out somehow. Either you do the teaching in vernacular, okay, or that you change now the you teach in a language like English or God knows what else. Decide that and then make sure that the teacher knows how to express herself or himself. But now at the same time, you have a problem of students coming from different parts of the country. Okay. Their perception of what you're saying also depends on what they have, their past history, linguistic history, from their schools, their childhood, their family, their relatives, their friends, whether they were coming from cities or villages, etc. It's a complex problem.
So a lot of the science teaching probably now has to be experiential. It should probably go beyond language. And I think
Yes,
when I was attending your lecture, and a lot of it is excellent, very elegantly designed experiments. So the experimental, way of understanding and experiencing it as you disrupt a system and try and understand what the system is trying to tell you. This is such
that's
great
right.
fun. But I think in our system, we do not encourage people to fail. So, trying things out, playing around with things, and, stumbling upon something is not encouraged. So,
Yeah.
one hears all the stories and, science and innovation, and that
Well,
scientific temperament is
yeah.
what we would like to inculcate in our future generations. But I don't know how successful we are being. So,
Well,
definitely, we have to change that.
the point is whether it's science or other subject, teaching is a storytelling process. You make up a story, you tell the story, and stories are convincing because no matter which religion you are of, you have mythological brain embedded, and one has to utilize that faculty to get the message across. And there's nothing wrong with it. It is not to consolidate their mythology, but use that as a technique. So teaching is a storytelling scenario. One should make up
Which,
stories, and
which
then they appreciate it.
brings me to splinters, your splinters. And
Yes,
when I was reading the cover, so it seems like you have sort of woven, the melody of poetry with the reality of science, and
that's
they are meeting somewhere in the middle. So I was talking about experiencing science. So would you like to end with maybe splinters? Some of your thoughts, anything.
oh, you want me to say something about splinters?
Yes,
Well, I'll
please.
tell you.
I was very
Okay, I'll tell
intrigued
you one
about it.
story of the. I'll tell you one story which you must have read. That's about when I was doing my doctorate in Geneva at night. As my experiments is to chick embryo experiments last 24, 36 hours. So at night I will take off from the laboratory and walk to the lake, Geneva Lake, and I saw this beautiful fountain. It's called Rocket Fountain in Geneva. It's called Jet of Water, Jet d'eau. So I looked at it and then I wrote a poem about it. What I was seeing in the jet of water, how the water comes in, how it spreads and sprinkles, and how when the light, because there were some at night there are lights around it. So how the lights make the particles shining particles, and if somebody is walking underneath, how the person may get wet, and all kinds of things. This is what I describe. This is what happened. This is experience as you live it. So you look at
Is
the
there any
poem Jet
particular
d'eau.
poetry on
If
genomic
have you
stability
Do
and
you have
cellular
the
aging?
collection, or do you
Not
have the
yet,
collection?
not yet.
You know, okay.
I will.
Maybe I'll mail you that poem. Okay.
So,
Then you can
is
take a
there
look. And
one
there are
on
many
genomic
such things.
stability? Is
Pardon
there
me.
one on genomic stability and cellular aging?
Yes. Oh, there are difficult, but no, I must say that in there my science was not affected much. The science, because I read a lot about astronomy also, not astrology, astronomy. I'm always thinking about perhaps establishing a colony of like-minded people on the Saturn, for example. Okay, and so there are things like that. So I have written about that also in one of the poems. Then I do a lot of games, word games, games of words in my poems, fishing for different kinds of scenarios. I write haiku, for example. Haikus are three-liners in. Then I write what are called four-liners also. Then I write long poems. Like there was a story about. I made up a story of a devil met uh, Devil met a lady who was uh, how do you call them? Forgot how to say that. And there's a dialogue between devil and this lady, and the devil is trying to persuade her to do a lot of things, and she's all the time coming back and shooting back to him, telling him, "No, if I do that, this will happen. So you devil, you're trying to pull my leg." So he keeps. So that dialogue goes on. There are things like that. The whole poem is like that. So there's a lot of variety because
Right.
these poems were written from 1960s all the way to 2010, 2015. So there are different times. Some of them were written in Pune, some written in Geneva, some in Lausanne, some in Okrishna, some in Paris, and depends where. Or there was even one I wrote in Tokyo, and it all
So
depends
these are
where I was.
little capsules of time of your feelings of a certain moment captured in
Yes,
words.
yes.
So, on that note, we thank you once again for
Yeah.
giving such a wonderful lecture, for sharing your experience. Most importantly, it has been a
Thank you.
privilege.
So
So,
pleasure
please
is
take
mine.
care, and I
Yes.
hope you will come back and again speak on our
Yeah.
webinar.
Incidentally, if you want to have a one on phylogeny, I mentioned it to Saili. I have a, a topic I prepared for some time now. It's called Darwin's Dream. Okay, when Darwin proposed phylogeny and evolution, what he meant to see, which I call Darwin's
Oh, that
Dream,
would
and
be excellent. So,
so
we will
molecular
schedule
phylogeny
another talk
as
very soon.
yeah, right. Okay. So that will be
Yeah,
interesting to geneticists also. That phylogeny based on single traits is has meaningless, but
right,
phylogeny must be based on multiple traits, and that's what Darwin trapped. When he looked at
right.
the whole animal body structure and compare an elephant to a cow and a man, he looked at the whole the totality as he could see it without having to go inside, but he was looking at multiple traits, and which is not what molecular biologists do. They look at, take actin, for example, or tubulin and make a phylogenetic tree of
Mm,
tubulins and actins. Has no meaning because every
true reductions,
phylogenetic tree for a different protein looks different. So anyway,
indeed.
that's what
So
Darwin's dream is about.
that is another lecture that we all look forward to. With that, we
Okay,
thank our
thank
speaker
you,
and we thank our audience tonight and the future audiences who will be tuning in to your talk and this discussion, which will be archived
thank
on
you
our
very
channel.
much.
So
Thank you
thank
for
you
the
so much.
opportunity.
Take care and bye bye.
Bye, bye, bye. Thank you, Saili.
Thank you, sir.
Right. Bye.
And lastly, thanks to our audience who for your patience. And if you like, you can like this session, and you can share with your friends and colleagues and subscribe this channel, and also join us on next session, which will be on 20th March and 21st March, next weekend. So till then, stay happy and stay good. Thank you.